Anatomical Enablers and the Evolution of C-Force Photosynthesis in Grasses examines the reasons why some plant species have the tendency and capacity to help facilitate the evolution of C4 photosynthesis. Before diving into the findings of the paper, let us first address the question of, what is photosynthesis? Photosynthesis is the process in which autotrophs harvest light energy to be converted into chemical energy. The wonder of photosynthesis primarily stems from the ability to self-sustain using just sunlight, carbon dioxide, and water. Together, these inputs combine to form energy stored in the chemical bonds of carbohydrate molecules as well as oxygen gas. So how does photosynthesis work? Light energy is captured by chlorophyll, which is then converted into chemical energy in the form of chemical bonds that compose the carbohydrate molecule. The net chemical equation for photosynthesis can be seen below. However, it is important to note that photosynthesis is really accomplished through two linked reactions. The first reaction is a light capturing reaction, which converts sunlight into ATP and NADPH molecules, as well as produces oxygen gas. Next, carbon dioxide is reduced in the Calvin cycle to produce a carbohydrate molecule, fueled by ATP and NADPH from the previous reaction. Here is a diagram that shows the two linked reactions. Now we know enough background to distinguish between C4 and C3 photosynthesis. C3 photosynthesis produces a 3-carbon molecule, while C4 photosynthesis produces a 4-carbon compound as an intermediate during its two-step process to produce carbohydrate. The advantage of C4 photosynthesis is that it allows plants that live in dry, warm, and sunny conditions to concentrate carbon dioxide around its chloroplast. By doing so, photosynthesis becomes more efficient with the ill effects of photorespiration eliminated. With this basic knowledge of photosynthesis and the difference between C3 and C4 photosynthetic plants, let's take a look at a peer-reviewed journal examining C3 and C4 difference in grasses. C4 photosynthesis is more productive than C3 photosynthesis because of the foliated anatomy within the C4 structures. It has been shown that C4 photosynthesis takes place in 22 to 24 grasses, all within the PACMAD clade and none in the BEP clade. The question addressed within this study is, why does this clustering happen? What are the evolutionary reasons? What mechanisms allowed for C4 photosynthesis to take place? The overall procedure of the study was to analyze the cross-sections of grasses in order to construct a phylogenetic tree with the hope of understanding the relations between the clades. Data was collected from 157 different species of grasses within the PACMAD, BEP, and outgroup species. The examination of the cross-sections yielded data about the bundle sheath cell size, distance between the cells, and thickness of the outer and inner sheaths. The results of the analysis were compiled and a phylogenetic tree was created. This is the phylogenetic tree presented in the paper. This graph shows how the grasses tested are related to one another. This is a simplified version of the phylogenetic tree that specifically shows when each trait evolved that correlated to the development of C4 photosynthesis. Initially, both types of grasses had a reduced distance between the bundle sheath cells. This trait allows for the carbon dioxide concentration to increase, which escalates the rate of photosynthesis. However, as the two grasses clades separate, the size of the bundle sheath varies. The study showed that the combination of large bundle sheath cells and a reduced distance increases the chances of C4 evolution. Essentially, the clustering of C4 photosynthesis occurred in the PACMAD grasses because of the combination of traits that allowed for a foliated anatomy. It is believed that when carbon dioxide levels decreased 30 million years ago, plants such as grasses adapted by concentrating carbon dioxide around chloroplasts to aid in photosynthesis. This led to decreased cell distances in the bundle sheath and the eventual evolution of C4 photosynthesis.
Now that we've taken a look at Chris et al.'s paper, let's take a look at the big ideas involved in the research. This research team built a phylogenetic tree for the BEP, PACMAD, monophyletic group, and rationalized their findings with the concepts of natural selection and evolution. By taking both the phylogenetic tree this team created and the characteristics of C4 plants into consideration, we can see that the BEP clade does not have C4 raised structures while the PACMAD clade does. These C4 traits include short bond sheath distance, large outer sheath, inner sheath cell areas, high outer sheath, inner sheath cell area proportions, and C4 enzymes, which are present in most angiosperms. After this monophyletic group split, the PACMAD clade developed more C4-like structures, while the BEP clade developed more C3-like structures. Now let's look at the phylogenetic tree that demonstrates this. This phylogenetic tree supports the idea that anatomical differences led to PACMAD developing C4 structures much more easily than the BEP group. Some selective pressure must have pushed these groups to diverge. In looking for this selective pressure, the team rationalized that before the split there must have been no selective pressure for CO2 fixation efficiency, and thus no need for C4-like traits in these plants. The team concluded that after the lineage split, CO2 levels decreased, which pushed for greater CO2 fixation efficiency and thus C4 pathways. When CO2 levels dropped, C4 pathways for photosynthesis carried a fitness benefit over C3 pathways. The PACMAD clade was pushed to become more C4-like, while the BEP clade was unable to develop C4 pathways. In comparison to C3 plants, C4 plants can make use of more carbon for fixation, as the C4 pathway acts as a CO2 concentrator. This increases the efficiency of photosynthesis in hot, dry conditions. In the PACMAD group, C4 traits became more pronounced as the fitness benefits of efficient photosynthesis increased.